susceptibility test that we are commercializing in collaboration with Mayo Clinic Laboratories. And the company now has about 80 employees, and we've treated over 50 patients on a compassionate basis. We've raised over $110 million from investors and also mostly from U.S. Department of Defense contracts, which is a lot of money, but it's a very expensive process. So we're going to have to raise additional money before we actually get our Phage products to market. Our investors include Deerfield Management, which is one of the largest venture capital funds in the anti-infectives space, and the AMR Action Fund, which is a recently organized venture fund, which includes funding from most of the large pharmaceutical companies. They have $1 billion, and we were their first investment. And then the Mayo Clinic has also invested, as well as some other healthcare systems and private investors. The company is located near Washington, D.C., which just happens to be where I grew up. But there are strategic reasons why it's good to be located near the nation's capital, because we have easy access to our FDA. We also have we're close by the National Institutes of Health, and both the Army research facilities and the Navy research facilities. So just to give an overview of how we see our Phage bank working, we see two basic use cases. One, which I'm going to talk about primarily, is a personalized use of a Phage bank, in which a patient with a chronic infection comes to the hospital. Their culture is tested against our collection of Phage through a Phage susceptibility test, or PST. The pharmacy at the hospital has an inventory of perhaps dozens of Phage which have been manufactured and have cleared FDA allowance to be used as a drug. And based on that Phage susceptibility test, the Phage is administered to the patient as personalized therapy. And this process takes currently about 5 days from the culture from the patient to delivery of that personalized Phage therapy, which that works for more chronic infections, but obviously has challenges for acute infections. So the second use of our Phage bank is for the more acute infections. And that's where we look at the epidemiology of the causative pathogens for a particular disease, like pneumonia or bacteremia. And we're able to look historically at which Phage would have been able to lyse the bacteria that was causing those infections within a particular region, and then select those phages as a mixture to deliver them immediately to the patients. So that's the second use case for the way that we see these Phage banks being used. The reason why we're all interested in Phage is because antibiotics are beginning to fail. We're seeing that when an antibiotic is launched in the market, the very first day the antibiotic is available, it's at its best range of coverage. But over time, the use of those antibiotics drives the selection of the resistant bacterial strains, and soon all the bacteria become resistant to the antibiotics. And of course, this has been happening over and over again since the very first antibiotics were introduced in the 1940s, and it's causing the antimicrobial resistance crisis. Phage therapy, which started before antibiotics were discovered, has been challenged in a couple areas, but one of the main areas has been in the narrow host range of individual Phage. When the United States was involved in the war in the Middle East, in Iraq, soldiers were coming back from Iraq with Acinetobacter baumannii infections that were not responding to antibiotics. And because of that, the US Department of Defense started looking at phage therapy as an alternative to antibiotics. And they turned to this concept of a phage bank. And my father's work at NIH played a role here because one of his postdocs was hired by the US Department of Defense to head up this effort to build this phage library to address this problem. The result has become the APT Phage Bank. And it was, as I mentioned, it was originally envisioned by my father, Carl Merril, when he was at the National Institutes of Health. And we were able to acquire, in the starting of Adaptive Phage Therapeutics, the collection from the Navy Biodefense Lab. And more recently, we were able to also acquire the collection from the Walter Reed Army Institute of Research and their phage collection. We've also partnered with a number of academic phage labs to acquire their phage collections. And we built a pretty large lab here within Adaptive Phage Therapeutics for our own phage discovery. So now we have hundreds of different phage with in vitro activity against all of the AMR high priority bacterial species, bacterial species. So after we started the company, actually before we even started the company, the Navy lab got a call, and many of you maybe have heard of the case of Tom Patterson. That case kicked off this whole phage reemergence of interest. This is the actually a slide on this first case. That's Tom Patterson and his wife Stephanie Strathney. They went on vacation in Egypt on a river cruise down the Nile, and Tom contracted an Abumani infection and became critically ill. He had been in a coma, and there was no expectation that he was going to survive. Fortunately, his wife Stephanie has a PhD in infectious disease epidemiology. She's on the faculty at UCSD Medical Center. She knew about phage, and she reached out to that Navy Biodefense Lab to ask if they would use phage to try to rescue her husband, which they agreed to. And now that's become a famous case. And it was the world's first use of intravenous phage for treating a systemic Abumani infection. After that case, and Stephanie's been wonderful at drawing a lot of attention to phage therapy, there's been a lot of interest, and we have been inundated, unfortunately, with many patients who have run out of options with traditional antibiotics and need phage therapy help. And we try to treat as many as we can, but it's a small fraction of how many are asking for help. On average, I'd estimate we see one request per day from a patient asking for compassionate use of this phage therapy. I'm just going to highlight a couple of these cases that were landmark cases that sort of illustrate the power of a phage library. This patient, which is our case number 7, was a taxi cab driver in Israel, in Jerusalem. He got into a fight, and his friend ran him over with his car, and his leg became injured and became infected with both Abumani and Klebsiella, both multi-drug resistant strains. After 7 months in the hospital on antibiotics, you can see the image there, his wound was not closing, and the clinical standard of care was to amputate his leg. The treating physician reached out to us to see if we could use phage therapy in an attempt to save his leg. They sent us a culture of both species, the Klebsiella and the Abumani, and we sent back the matched phage for them to administer through an intravenous route. And after 2 weeks, the wound closed, and this patient was able to leave the hospital. It was the reason why I think this is really a landmark case is because this was a polymicrobial infection, which you could not have treated with a standard off-the-shelf mixture of phage. It really required a large polymicrobial phage library which fortunately we had, and we were able to match the phage to his particular infection. As a little side note, unfortunately, this patient passed away just a couple of years ago from COVID. So the treating physician, Dr. Nirpaz, he made the comment to me that a virus saved this patient's life, and then a virus took the patient's life. On a more happy note, this patient, John Haverty, had his knee replaced about 12 years ago, and the prosthetic joint became infected. He was being treated at the Mayo Clinic, and they operated on him to try to clear the infection. They ended up doing 17 surgical procedures over an 11-year time span, and the infection would not be cleared. So he was looking at amputation as his next therapeutic course. And unfortunately, because of all the previous surgeries, the amputation would have had to be at his hip, and he would have to be constrained to a wheelchair. The Mayo Clinic asked if they could try the phage therapy, and they sent us a culture. He also had Klebsiella infection, and we were able to match his bacteria to phage within our collection. And they administered the phage through an intravenous infusion. And within 48 hours, there was no sign of infection. The pain went away. He could bend his knee. And it was the world's first use of phage in lieu of surgery to treat a prosthetic joint infection. It was so dramatic for the Mayo Clinic doctors that the Mayo Clinic called, and they became an investor in the company. And they partnered with us to advance the phage susceptibility test through their laboratories, which are quite large. They're in the United States, they're the third largest diagnostic testing labs. So it's a wonderful partnership. But the big challenge for us is, how do we translate those experiences into the necessary regulatory randomized controlled trials that are necessary for licensure and broad market availability? So one of the things is that in the emergency use, our phage matching is challenging to scale. And that's one of the beauties of teaming up with the Mayo Clinic. And we've developed our assay now so that we're looking at both the planktonic activity of the phage against the planktonic bacteria, but we're also looking at bacteria that exist within a biofilm and seeing if the phage can eradicate the bacteria even in the presence of the biofilm. And then we're also looking at the use of the phage adjunctively with antibiotics, and making sure that there's synergy between the antibiotic and the phage. And so our phage susceptibility test is looking at all of these parameters, and we're trying to pick the appropriate phage for that patient. That phage susceptibility test acts as a way for us to match the phage for a patient, but it has a second purpose. And that second purpose is that by constantly doing this testing, we're able to have a good surveillance of which bacteria are in the environment and which phage match that bacteria. And if a bacteria presents itself that we don't have phage for, we're able to capture that bacteria and put it through a process of additional phage discovery. So we do our coverage expansion. And this process takes about 4 months for us to complete for when we have a new resistant bacteria that's been identified. The first step is the phage hunting. Typically, that takes us just a couple weeks for us to find a new phage from environmental materials, given the pretty significant resources we have for phage hunting, in addition to our collaborations with the biodefense labs and our academic partners. We then do genetic screening. We do biobanking. We do our GMP manufacturing and our quality control release. So we have broad availability of that phage. within that 4-month time frame. Within that process, there are 2 steps that involve regulatory submissions and review. The first step is the submission of the genomic information on the phage that we found and our annotation and analysis to show that there's not deleterious genes, toxin genes, or genes that could support antibiotic resistance. We look for these things and we submit that information to the FDA through a drug master file, and I'll talk about that in a little bit. The second submission to the FDA follows the manufacturing. After we've manufactured the phage, to our negotiated levels of purity, we submit that purity analysis to the FDA ahead of being able to use that phage in a patient in our clinical trials, and hopefully when we get to commercial launch to make it broadly commercially available. So that those phage that pass that regulatory review gets added to the collection and is immediately available for use for future patients that will test for that pathogen that has that sensitivity. The most profound aspect of that whole cycle, that closed loop of surveillance followed by phage discovery and manufacturing, is that if a new bacterial strain is identified that is resistant, we can have broadly available a therapeutic agent within 120 days to treat a patient. And when you compare that to the traditional antibiotic development pathway, it would take 10 years, usually 10 to 15 years, to produce a new antibiotic and get that new antibiotic on the market. So this is a huge paradigm shift in our ability to address antibiotic or phage-resistant strains of bacteria. And what that would look like if you compare it to that original graph that I showed you, where a new antibiotic is introduced, and every day after it's introduced, it loses its ability to cover bacteria. With this method, the coverage that you saw on day 1, when resistance is discovered, resistance to phage, that is, we can add phage, and over time, you don't lose the coverage range. In fact, you actually increase the range of coverage over time. It's the first approach to antimicrobials that increases its spectrum of coverage. That's one of the things that I think is very exciting. So just not to be discouraging at all, but I just want to share with you the level of effort that it's taken for us to develop and advance this approach. So even for these early stage clinical development, it is a significant effort that spans manufacturing, building this facility. You can see that it's a fairly large facility that we've built here in outside of Washington, D.C. The quality control, the process development, clinical trial operations, our regulatory affairs teams. So I'm just showing you the organizational chart to just let you know the scope and scale of what we've built to try to support these clinical trials and advancing this phage, adaptive phage bank approach. Here you just see each of those squares represents a person that we currently have employed to help with our manufacturing activities. And that's just the manufacturing. Then you have an entire team that's responsible for ensuring the quality control, the quality assurance, the quality control. It includes doing the assays on the materials, analyzing the genomics, and also doing quality control over the clinical programs. But the quality control team is almost as big as the manufacturing team. And then we have a whole team that's developing new assays for doing quality control, as well as developing assays for formulation, or just developing formulation, developing stability, testing. and doing the phage hunting as well. So that's a smaller group, but it's an activity that's going on here. And then running the clinical trials. We have from our chief medical officer down through our vice president of clinical operations and the people that are involved with statistical analysis and running the clinical sites. And these are just the people that we have on staff. There's also a significant number of people that we have through CRO or contract research organizations that help us run the clinical trials. This is the most expensive part of the business, is running these clinical trials and organizing these clinical sites. And then we have a regulatory affairs team. And there's also people at the CRO as well that help us with regulatory affairs, and then some consultants as well. And just finally, to step through the 3 clinical trial areas that we're currently working in. We are advancing a clinical program for prosthetic joint infections, which are very difficult to treat with current standard of care, because these infections often involve biofilms, and they're in areas in the synovial space in the joints where the antibiotics are not very effective at even getting to those sites. So we really leverage the ability of the phage to replicate at the site of the infection, and the ability of the phage to work in the presence of biofilms to help with these infections. So our studies we have a study that is a randomized placebo-controlled study for patients who have chronic prosthetic joint infection. These studies are currently all in the United States, at clinical sites in the United States. And then we're also doing a study in patients who have diabetes, who have infections in their feet where the infection has spread to the bone. And also very challenging for traditional antibiotics to deal with these infections. But we believe that the ability of the phage to replicate at the site of the infection will make it better than many antibiotics. This is also a randomized, placebo-controlled, multi-site study. And then we have a study that we're doing in collaboration with a group called the ARLG, which is the Antibiotic Resistance Leadership Group, which is a nonprofit group of infectious disease doctors, and also funded by the National Institutes of Health. And this study is a study in cystic fibrosis patients who have Pseudomonas colonization, asymptomatic, so they're colonized, but they're not considered to be sick or have an active infection. And we're looking at a single intravenous infusion dose and the effect of that phage administration on the microbiological clearance within these patients' lungs. So those are the 3 active programs we have here. And just as a final slide to sum up the regulatory interactions. So in the United States, phage therapy is regulated by a group within the FDA, which is the Center for Biologics. All other antibiotics have always been regulated by the Center for Drug Evaluation and Research, or CDER. But CBER deals with biological entities and traditionally has been the group that has regulated vaccines. That's been a real benefit for us because they're familiar with ways of regulating vaccines or drug products which change. They have been managing the annual flu shot, which every year they're able to update the flu shot without having to do large clinical trials. So we're leveraging some of those lessons learned within that group at the FDA. And one of the things that they use is a mechanism called a drug master file. The drug master file allows us to upload the genomic information in our analysis of the sequences of the phage to the FDA on an ongoing basis, so they can review those on an ongoing basis. And then they can also review the certificates of analysis after our manufacturing to evaluate the purity of our products. And they allow us to add those phage to our collection, even in the middle of the trial. And we hope that in success with our clinical trials, that the FDA will continue to allow this process to go on so that even at commercial scale, we can consistently be adding phage to the collection and therefore overcoming any emergence of resistance to the phage that the bacterial populations will have. So the two areas that the FDA is looking at for us are the manufacturing areas, the CMC areas, and the clinical areas. Our product is a library of phage. In some ways, though, the way that the FDA is regulating this collection is by regulating our process for manufacturing and a process for clearing these phage. So that's not technically what they're doing. Technically, they don't regulate processes, they regulate drugs. But in effect, they are reviewing making sure that our process is working for this phage bank approach. As I mentioned, all the phage have to be screened in terms of looking for deleterious genes by doing the sequencing of the phage. And then they look at the purity through that drug master file. And the purity is related to things that include the host cell contaminants and endotoxins and exotoxins. And the rates for that have either been previously established by the FDA or have been part of a negotiation between us and the FDA. I think a key point that I highlight in the slide is that the ability of the phage to be added to the collection is, I think, a key point. And my understanding is that some other regulatory authorities in Europe, for example, aren't yet to the point where they would allow new phage to be added to a collection like this. But I think it's critical that the regulatory agency continues to allow us to add phage, because that's the only way we're going to break the broken development cycle of antibiotics, is by constantly adding phage with the emergence of resistance. And then on the clinical side, the FDA, after 100 years of phage being, phage was discovered, over 100 years ago, the FDA has confidence that phage is safe. And so they told us that we don't need the preclinical toxicology or PK/PD studies in order to start our clinical studies. And further, they also told us that it would be a waste of our time just to do safety studies. And we should really include efficacy signals in our first clinical studies. That's different than many antibiotic studies, because many antibiotics have toxic side effects which require safety studies. So phage has a real advantage in its known safety profile. So the FDA has told us that the initial studies, so we can look at efficacy. Now, having said that, these initial studies, the FDA, in allowing a study to go forward, their primary concern is to make sure that the product is safe. And they do believe phage is safe. But they're not as concerned about whether the product works for the initial studies. From the FDA's perspective, from a regulatory perspective, the initial studies, safety is the most important thing. And if it doesn't work, that's really at the risk of the sponsor. But when you move toward the later stage programs, the pivotal trials, then the FDA starts focusing on safety and efficacy and the ability of the sponsor to manufacture the drug product consistently. So that's my presentation for you today. And I appreciate the opportunity to talk to everyone, and I hope everyone's excited about phage therapy as I am.